presentation on social behavior in mice. Okay. So just to explain a little bit about what that actually means, so animals need to adopt certain behaviors that address their most urgent needs, but sometimes these needs might conflict, so we need to prioritize the most important. So psychologist Abraham Maslow described this as the hierarchy of needs, where our most basic needs like hunger take precedence over our social needs. And an example of a social need is empathy, and empathy can be defined as sharing the emotional states between individuals. So you can think of empathy as state matching. So when two babies cry, or when a baby cries from hearing another baby cry, or when yawns are contagious. And we see this in animals as well. And so in our lab, we study the social transfer of pain paradigm that studies empathy in mice.
So the main goal of our project is to use the social transfer of pain paradigm developed by our lab to study how hunger affects empathy-related and pro-social behaviors between a bystander and its injured social partner. And we want to do this through an unbiased lens using computer vision and machine learning tools. So in this model, a bystander mouse is socially interacting with a familiar mouse that's experiencing pain due to a hind paw injection of an inflammatory mediator such as capsaicin. And after the social interaction, we observe that the bystander mouse has an increased sensitivity to pain that mirrors the pain state of its injured partner. And we want to see how hunger affects this process. So we food deprive the bystander mouse for 24 hours before the social interaction. And what we found after it interacts with its pain mouse is that the bystander mouse doesn't adopt the pain state of its injured social partner, suggesting that food deprivation inhibits the social transfer of pain.
And so our main two questions were, what behaviors occur during the social transfer of pain, and then how does food deprivation of our bystander mouse affect these behaviors? So here's just a pipeline of our data analysis, and once again, we were really keen on trying to extract and quantify these behavioral metrics in an automated and unbiased approach. So we first started with the video acquisition. So we acquired high quality 30-minute videos of the social interaction between the bystander and the pain mice in a bottom-up perspective. And here's an example. So on the left is the baseline video of our control mice, where they appear to be independently roaming around the enclosure. And then on the right here, we have our social transfer of pain paradigm, where we begin to see a shift in how the two mice are interacting with one another.
And from this, we want to be able to track the positions of the mice throughout the entire video. So we use SLEEP, which is an open source deep learning framework that we use to extract pose estimation data. And we trained a custom model for our specific video training set. And we ran through this iterative process over the course of several months and trained on over 900 frames to be able to predict on our experimental videos. So on your left, you can see SLEEP track the two mice pretty accurately as they're exploring their enclosure. And on the right is during the social transfer of pain paradigm, where SLEEP, our model, is still able to preserve their unique identities even when they're closer in proximity and engaging in pro-social behaviors.
And so after we extract that pose estimation data from SLEEP, we applied two different behavioral analysis strategies. So we just first started with general Python script in order to capture the simple behavioral metrics between the two conditions. So we looked at total distance traveled and average velocity. And then we used key point mosaic as a tool to classify the behaviors of our food deprived bystander mice. And this allowed that automated and unbiased approach. And so more into key point mosaic. So it's an unsupervised machine learning tool that was developed by Bob Data's lab. And basically it finds these reoccurring patterns of movement that it defines as behavioral syllables. And this is all without any human input. So the way that it works is that we input key point tracking data from SLEEP, and then our modeling takes two phases. So we start with putting an initial model that breaks our data into discrete states so we can see how the behavior evolved over time. time. And then we fit a full model on all of the key point data, and this allows us to get those reoccurring distinct behavioral syllables. And this is just kind of like an example of what a syllable would look like.
So we'll now go into the results of what is specifically happening during this social interaction between a regular bystander mouse and its injured social partner. So here are example trajectory plots of the two mice as they're exploring their enclosure. On the left is from our baseline activity before any painful stimulus is administered to the pain mouse. And on the right is after we injected capsaicin into the hind paw of the pain mouse. And even at a glance, you can see a very distinct change in patterns of behavior compared to the baseline. Specifically, we see that the pain mouse is huddled in the corner, which is expected given its injury. And interestingly though, the bystander mouse is next to the pain mouse and is investigating or just in general closer to the pain mouse in general. So based on our previous observations, we then wanted to test our hypothesis by seeing if close proximity is the specific discrete behavior that drives the social transfer of pain from the pain mouse to the bystander. So we measured the distance between the two tail-based nodes as, I guess, an evaluation of how generally close they were together during the social interaction. And to our surprise, there was no significant difference from the baseline to the post-pain injection.
So we looked at some other metrics. So we looked at average distance traveled and average velocity, and here we found that the bystander mouse was traveling a significantly shorter distance and moving at a slower speed compared to our baseline groups. So that suggested that the administration of a painful stimulus was not only modifying the behavior of our pain mice, but it was also modifying the behavior of the bystander mice as we expected.
So we'll now go over the results using the same methods to understand how food-deprived mice respond differently to an injured social partner compared to their fed counterparts. So here are the same trajectory plots as before, and we're now comparing a regular social transfer of pain paradigm to one where the bystander has now been food-deprived. And in the regular fed group, we see that the bystander mouse follows the trajectory generally of its pain mouse, which aligns with what we saw in the previous trajectory plots. But in contrast, the food-deprived bystander doesn't tend to stay next to the pain mouse and is instead exploring all over the enclosure.
And so now we're looking at just our social transfer of pain paradigm, and it yielded the similar results as we showed earlier. But now that when we look at the bystander or food-deprived bystander group, we see that the food-deprived bystander is traveling at a longer distance traveled, and then it has a higher average velocity. So this might suggest that the food deprivation is actually inhibiting the social transfer of pain for our food-deprived bystander.
And also looking at average distance apart again between tail-based, we found that the food-deprived mice were in general significantly farther away from their respective pain mouse compared to the fed group. And this aligns with our hypothesis and possibly suggests that in a state of hunger, the mice are shifting their priorities away from social interactions and more towards resource seeking or self-preservation.
So now let's just transition into our key point most-seek data. So we use key point most-seek to find those reoccurring distinct behavioral syllables, and it goes by frequency. So 0, or the syllable 0, is the most frequent syllable, and then syllable 12 would be among the least. So this is just an example of our data set, and we can see a lot of walking and turning, jumping, running. So this is suggesting a lot of hyperactivity and exploration happening during the social interaction. So this is just a trajectory graph. So the nodes are representing the syllables, and then the edges are representing the transitions from one syllable to another, and then the width of the edges are the transition rate. So for example, syllable 0, which is walking, transitions frequently with syllable 4, turning, and then syllable 1, rearing, transitions frequently with syllable 9, running.
So in future analyses, we hope to compare what we got here from our food-deprived bystander and run the same data analysis on fed bystanders to see what behavioral syllables are most common in that group so that we can compare the two and really see what discrete behavioral differences are happening between the fed condition and the food deprived condition. So, in summary, we set out to explore how animals can prioritize competing needs, particularly the physiological drive of hunger and the psychological state of empathy. And we use post-estimation data extracted from sleep in order to visualize and quantify the behavioral changes that occur during the social transfer of pain. And this allowed us to move beyond qualitative observations and really quantify the changes that are happening between the two groups. We found that food deprivation significantly changed the behavior of the bystander mice, suggesting that hunger may override empathy-like responses in mice through these discrete changes.
And we used Monique, which was an unsupervised machine learning tool to really extract and cluster these distinct behavioral syllables that allowed us for that unbiased and automated approach. So by integrating machine learning into our animal behavioral research, we automated our data analysis pipeline, and we also decreased the labor intensiveness of behavioral scoring, which we typically have with human scoring. And we also got new insight into how complex social behaviors are modulated by competing biological needs. And these are our references.
And thank you so much for listening to our presentation. We'd like to give special thanks to Dr. Monique Smith, who is our PI and developed the social transfer pain paradigm, as well as our graduate student mentor, Amanda Nguyen, who ran the food deprivation experiment and allowed us to use her data for our presentation.
Thank you.
Thank you.
We have just a minute left. Would anyone like to ask any questions to our final presenters? Or offer comments?
Yes.
I thought your presentation was really interesting, and I know that you brought up a couple variables like pain and hunger. I was wondering if you guys saw any insights on the differences between sex within your analysis too.
So, for the food deprivation specifically, we saw that only female mice exhibited this change in empathy-related behavior. But in the general social transfer pain paradigm, we saw the same results in both male and female mice.
Thank you.
How about another round of applause for all of